to Spiritual Dessert Truths, episode 106 for Russell Brand and other people, continued about the compatibility gene. So we're on the topic of sex and smelly t-shirts again. We're still in this experiment that was quite interesting with sweaty t-shirts. And it seems that the statistics are we go for the gene that is the most directly opposite of ours so that you have the greatest range, of course, natural selection of incompatibility. So this way you have the largest range of immunity. They were giving him a hard time because they thought that it's not fair. Referring to smells could vary. They really don't know about smells. They're going back to the t-shirts. He tried storing them in the freezer before being smelled so that maybe it's a little bit more, the smell is lost over time upon freezing. Each additional step would have a bigger effect. And in the more recent experiment, women first smelled all the t-shirts before scoring each one separately, whereas in the Wittekind's experiment, women scored each t-shirt on the first encounter. Nobody knows if this subtly influenced the outcome, but in the 2008 study, however, revealed a small increase in desire for men with similar compatibility genes for women taking the contraceptive pill, which was in agreement. So apparently it throws you off. If you're on the pill, you're not selecting properly. Fortunately, I'm not. Makes you more attractive. Makes you dangerous. That's what it does. Even so, conclusions about the use of the pill remain debatable. It directly changes women's preferences. It could also relate to other factors linked with using the pill, such as one's relationship style or status. Another experiment, 2002 in Chicago, came to the startling conclusion that women's own compatibility genes influenced her preferences differently according to which genes she had inherited from mom or dad. The method for this experiment was familiar. T-shirts were worn by men for the 2 days and then scored by the women. One conclusion from the study was that there wasn't any male smell, that all or even most of the women liked or disliked. Preferences varied. This is one of the reasons why students and politicians needn't have panic when they first heard about his plans, because these studies show that diversity is what's important. There's no superior smell. Everyone gets preferred by someone. But the unexpected discovery from the study with Hutterite women was that they preferred the smell from men who had similar compatibility genes to their father. Preferences didn't correlate with their mother's genes. Women liked men who smelled like dad. Our eminent scientists commented that with this discovery, the influence compatibility genes have on behaviors rivals their importance in immunity. These results seem to be the complete opposite to Wittekind's earlier experiment, which found that women preferred dissimilar genes to their own, just like others had done to him all those years before. Wittekind published a note complaining about the way that the statistical analysis was done in that study. In truth, these different results do not necessarily mean that one of the teams got it wrong. Behaviors influenced by compatibility genes have several different roles: avoiding inbreeding, identifying close family to be cared for, and maintaining diversity in the immune system. So whether or not a smell is regarded as pleasant, attractive, or sexy may well depend on the environment and of the atmosphere of the experiment, the way the question is asked, and the culture of the people involved. All aspects that are very difficult to take account of. It seems feasible that any one type of situation, women prefer the smell of t-shirts with men-related genes, while in other circumstances, those worn by men with different genes smell sexier. We all know that feeling sexy is much more mood-oriented, so these studies may have arrived at different conclusions if they might have been a full moon. Given each difficulties, perhaps working out how we smell each other's compatibility genes would shed light on the situation, and how something works is usually easier for scientists to tackle compared to why. One possibility is that HLA proteins, or peptides presented in their groove, are smelled directly in mouse urine. Fragments of these proteins have been found, and sensory neurons in the mouse nose have receptors that could mimic the receptors on immune cells that detect the proteins. Humans, HLA proteins are found in the blood, but they're not volatile and not easily changed. An alternative possibility is that compatibility genes may be sensed indirectly. Many scientists, including Whitaker, consider it likely that something volatile correlates with the versions of compatibility genes a person has inherited, and that's what can be directly smelled. Most of the research to work out precisely how odor relates to compatibility genes has been in mice. Because the use of inbred strains allows comparisons between mice, those poor mice, the different types of experiments have been done. One approach is to capture the volatile chemicals emitted from the mouse urine. Well, imagine that job. I don't think that's a good job for me. That's a torture. Forget it. I could smell it. I could smell it from here. Maybe I could go work there. The Manel Chemical Senses Center. Probably closed. We still don't really know how it works. After all that, humans and mice have a fixed number of compatibility genes. This behavior is very likely aimed at achieving an optimal inheritance of immune system genes. But it isn't simple as that. See, all the fish prefer partners with high numbers of compatibility genes. There's a balance that determines how many of these genes is optimal for the immune system. Something we discussed for humans in a chapter that we're not reading right now. We don't have hundreds of these genes. So no matter what, you're only going to select the most important ones, and that's, again, the most important part. A balanced immune system is best. And consistent with the idea, female sticklebacks, some kind of fish, with an already large number of compatibility genes, were less keen on the smell from the males who also had very many genes, and those with few compatibility genes themselves were keen on the scent of the males with the more. Makes sense. Balance. So, in fact, 6 ends up to be the number of these particular compatibility genes that the fish inherit most often. Benefits against malaria, sickle cell anemia, things like that. There's evidence that this is the case. Which one is it? For humans, it's commonly thought of that our sense of smell is relatively feeble in comparison to many other animals. It just goes back and forth, bouncing around. It's not fair. We know there's a specific odor for coffee, even though we don't even know exactly how it works. But it gives us that homey feeling. Even if compatibility genes are just one of the many factors that influence our relationships, the implications are profound for understanding human biology. It's even possible that the compatibility gene system first evolved to allow individuals to distinguish kin from others, and then later these genes were hijacked by the immune system to be used for the search for disease. Fundamentally, this research area expands how the human body can discriminate self from non-self, influencing how we unconsciously sense family members, strangers, or lovers. More research is sorely needed, yet this is just one hint that compatibility genes reach into human biology far more deeply than first thought. Next, your brain. Pleasant dreams. Nighty-night. Love you.